This video begins 24 hours after the induction of the experimental sunburn on the thigh of a patient using a calibrated UV light source. After application of local anesthetic, a microdialysis catheter is inserted between the cutis and subcutis and is then pulled through the skin. A perfusion syringe is attached to the proximal end of the catheter, and the activation of the perfusion pump will induce the flow and collection of microdialysate. Following collection, samples can be stored at -80 degrees Celsius until analysis of nociceptive and inflammatory mediators is ready to be performed.
Hi, I'm Martin Angst from the Department of Anesthesia of Stanford University.
And hi, I'm Martha Tingle, Senior Research Nurse and Manager of the Human Pain Laboratory, founded by Dr. Angst in 1995 here at Stanford University.
Today we're going to show you a procedure for the placement of microdialysis catheters in experimentally inflamed skin of human study participants for the collection of interstitial fluid.
We use this technique in our laboratory to study tissue-specific release of biochemicals critical for the mediation of pain and inflammation.
So let's get started.
One model of experimental skin inflammation is the artificial sunburn. A first-degree sunburn is induced with aid of a calibrated ultraviolet B source. Accurate delivery of energy is assured with a UV meter.